So let's start. So lecture 3, last lecture today, is about sodium balance. So why do we need to regulate sodium balance? The answer comes from the first lecture. The reason is that sodium is the most important osmotically active substance in the plasma, and as such, it basically is the most important determinant of your extracellular fluid volume. And if your extracellular fluid volume goes up, then eventually your blood plasma volume will go up. And if your blood plasma volume goes up, that's going to induce hypertension. And so in the long run, sodium balance is one of the most important determinants of your blood pressure. And so you want to regulate sodium because you want to avoid both hypertension and the other way around. You also want to avoid hypotension and shock. Because in modern societies, we have plenty of sodium, so avoiding hypertension is the bigger issue. But during evolution, avoiding hypovolemia was just as important, because if your blood pressure is low, then you're much less efficient in flight and fight. So since the glomerular filtration rate is very high, we filter 180 liters a day. That means because we have a lot of sodium in the plasma, we filter a lot of sodium, roughly 1.5 kilograms of sodium per day, which is more than 9 times the total sodium in the body. That's a lot of salt. It's like 2 boxes of Morton sea salt. So you need to use a lot of energy to reabsorb that sodium. And so we reabsorb more than 99% of it. And we only excrete somewhere around 1/10 of a mole per day, which is more or less similar to how much we take in. And other than the kidneys, you also have the sweat glands and your GI system, where you can also lose a little bit of sodium. But the most important regulator by far is your kidneys. So the regulation of sodium uptake is not as sophisticated and not as strong as the regulation of water intake. So thirst can be a very strong feeling. With sodium, we just simply love sodium. It's not very regulated. We love the taste. Any food that's good usually have some salt in it. These are the usual culprits. We usually eat far more salt than what we need. If your kidneys are okay, you're fine. But some of us can get high blood pressure from it. That being said, there is such thing as sodium appetite. So if somebody has low sodium content or low volume, people tend to crave sodium a little bit, but it's not nearly as strong as thirst, which can be really a very strong feeling. So what we're going to do in this lecture, I'm going to go over the different parts of the nephron. In each part, what happens to sodium. And there's going to be a lot of information, but this is important for many reasons. One of them is that all the commonly used diuretic drugs, which are used to treat hypertension, congestive heart failure, all of these drugs inhibit some sort of sodium transporter. And the other reason it's very important to remember all these mechanisms is because everything else after this lecture will build on this lecture. So almost everything is coupled to one way or another to sodium. And so this is a simple version of the nephron, which you already had before. And this is a slightly more complicated version of it in the context of the macroscopic anatomy of the kidney. You can see that the outer layer of the kidney is the cortex. This is this beige color here. And the inner layers are the medulla. These are these renal pyramids. And at the bottom of the medulla, you can see that these collecting ducts will collect the urine into the papillae and then into the renal pelvis. And then those collect into the ureter. What is also shown on this figure is that there are 2 kinds of nephrons. You have the deep juxtamedullary nephrons. This is the minority of the nephrons. These will be very important for water balance. And you have also the superficial or cortical nephrons, and these don't go to such depth of the medulla. And the importance of this going deep into the medulla will be discussed on the water balance lecture next week. So this is just a simple overview of what happens to sodium at the different parts of the nephron. So the proximal tubule reabsorbs roughly 2/3 of the sodium with 2/3 of the fluid. And so by the end of the proximal tubule, the fluid will be isosmotic or isotonic with the plasma. And the loop of Henle, these two thin limbs and the thick ascending limb, much of the sodium reabsorption in this part happens in the thick ascending limb, which is going to be more or less 25% of the reabsorbed sodium. And what is interesting here is that this is one of the very few places in biology where water will not follow sodium, because this part of the nephron is impermeable to water. And the reason for that, it doesn't have aquaporins. You will see this in the next lecture, next week, the water lecture. And the more distal parts, the distal tubule and the collecting duct, will reabsorb around 7% of the filtered sodium. And this is the part, even though it's quantitatively minor, but this is the part where the major hormonal regulation takes place by aldosterone. So again, everything builds on sodium. So this is where I'm going to briefly discuss epithelial transport mechanisms. There are two ways to move something across an epithelial cell layer. One is through the cells, which is called the transcellular pathway. What happens in a transcellular pathway is at the apical membrane. The apical membrane is the one that faces the lumen of the tubuli. On the apical membrane, whatever you want to move through the epithelial layer, in this case it will be sodium, enters the cell on some sort of a transporter of a channel and exits the cell on the basolateral side through another transporter or a channel. So transcellular, it goes through the cells. And again, if it's a reabsorption, it moves from the tubular lumen, from the apical side, to the basolateral side, which is the one that faces the interstitium and the peritubular capillaries. So this is a transcellular mechanism. The paracellular mechanism, on the other hand, goes in between cells, and epithelial cells are famous for being connected to each other by these tight junctions. And these tight junctions are formed by several different proteins, including claudins. And so in the paracellular transport mechanism, substances move through these in between cells through these tight junction proteins. And again, if you want to put this into the context of the tubuli, the apical membrane is the one that faces the inside of the tubule. And if you reabsorb something, you move it from the tubular lumen to the interstitium, from where it's going to enter the peritubular capillaries. Now, being somewhat more specific for sodium, the way this transcellular mechanism works is that you have the ubiquitous sodium potassium ATPase on the basolateral membrane of the cells. And you have, as you will see in the next couple of slides, a quite nice large variety of sodium transporting mechanisms, transporters and channels through which sodium will enter the cells in different segments of the nephron. And the transcellular pathway is very important because much of the hormonal regulation takes place here. And essentially all diuretics inhibit some sort of a sodium transport mechanism in the apical membrane. And as I discussed in the previous one, the paracellular route takes place in between the cells through the tight junctions. And this one is determined by the concentration gradient and the electrical gradient in between the interstitium and the tubular lumen. So let's start with the early proximal tubule. So the early proximal tubule has several mechanisms by which sodium is reabsorbed. One of them is using this sodium proton exchanger. The sodium proton exchanger basically moves sodium inside the cell and moves a proton out into the lumen. The thing with protons is that free proton concentration is very low, and it's basically the pH, a negative logarithm of free proton concentration is pH. So you can't really move a ton of protons into a solution without dramatically shifting the pH. So something always buffers protons, because otherwise you get enormous pH changes. And what buffers protons in the lumen of the proximal tubule is bicarbonate. Just like in the blood, bicarbonate is the most important buffer. And so you have roughly 24 millimolar bicarbonate in your plasma. That's the major buffer system. And that's freely filtered. It's a small molecule. So what happens is protons combine with bicarbonate and form carbonic acid. And carbonic acid will dissociate to CO2 and water. And CO2, being a gas, will simply diffuse to inside the cell. If you had pulmonary so far, you know that CO2 is very diffusible through biological membranes. And once inside the cell, it's going to join with water and form carbonic acid by the carbonic anhydrase. And the carbonic acid will dissociate to bicarb and proton, and the bicarbonate will leave on the basolateral side. This is a pretty complicated mechanism, but the end result is that sodium and bicarbonate together will be reabsorbed. Because basically, even though it's CO2 that diffuses through the membrane, the bicarb will regenerate from CO2 inside the cell and exit on the basolateral side. So this mechanism, where protons are buffered by bicarbonate, eventually will reabsorb sodium and bicarbonate together. We're going to repeat this one more time, because this is also part of acid-base balance by the kidney. So next week, we're going to have a lecture about regulation of bicarbonate. We'll come back to this point. This is one of the mechanisms. And what's important to know here is that the formation of carbonic acid from CO2 and water needs this enzyme called carbonic anhydrase. It does happen spontaneously, but it's very slow. So if you don't have the carbonic anhydrase enzyme, it's very slow. Also, it gets slower if you inhibit the carbonic anhydrase enzyme. And there are compounds that inhibit this enzyme. One of them is the prototype is acetazolamide. And acetazolamide was the first diuretic. Because if you take this drug, you inhibit the carbonic anhydrase enzyme, you slow down all this mechanism, and the end result is that you lose sodium. Now, acetazolamide is not the best diuretic, because you also lose bicarbonate, and that messes up your acid-base balance. So it's not really used, to my knowledge, as a diuretic anymore. But this was the first one among a long line of diuretics that has been developed. Now, this is one mechanism. The other mechanism in this nephron segment is co-transport with glucose. So glucose is a nutrient. You have roughly 5 millimolar glucose in the plasma, and you have roughly 24 millimolar bicarbonate in the plasma. So, you can reabsorb 25 millimolar sodium with this bicarbonate, and you can reabsorb 5 millimolar sodium with this glucose. And the way this works with glucose, there's this co-transporter which transports sodium and glucose together, moves it inside the cell. I would say the primary reason of this transport mechanism is to reabsorb glucose to make sure you don't lose this nutrient, but it goes together with sodium. So a little bit of sodium will also be reabsorbed by this mechanism. So now we come to the transepithelial potential difference, which is an important driving force for paracellular mechanisms. And what happens here, if you put an electrode in the lumen and put an electrode into the interstitium, measure the difference, what happens is you have a slightly lumen negative potential here. And the reason for that is because this mechanism is electrogenic. There's no counter ion, which means when sodium goes, there's no chloride going with it. So it leaves the lumen slightly negative. And this slight negative lumen will actually be a driving force to reabsorb a little bit of chloride through this paracellular mechanism. And one important thing that happens here also is that the chloride concentration will increase because a significant portion of the sodium here was reabsorbed together with bicarbonate. So if you remove sodium and bicarbonate, water will follow osmotically. So you leave chloride in the lumen, and this increased chloride in the lumen will be the reason why this transepithelial voltage will flip in the next nephron segment. So this segment is the late proximal tubule. And what happens here is you still have this sodium proton exchanger. You still have this sodium proton exchanger, but what happens here is at this mechanism, it's not bicarbonate that buffers the proton, but it's some other anions. And the reason why we don't specify what anion here is because the anion will just recirculate here. One of the anions that goes with this transport mechanism is formic acid. But again, it's not very important because it will recirculate, as you see. So what happens is this weak acid binds the proton, it becomes uncharged, and it diffuses through the membrane, and it's going to get, it dissociates the proton, and it becomes an anion, and it comes back to the lumen to this anion chloride exchanger. So basically, the anion recirculates, and the end result is reabsorption of sodium together with chloride. It's a complicated mechanism. I don't know why it's working this way. It's a lot simpler if it just was a sodium chloride cotransporter, but this is just the way it is. So basically, with the combined action of an anion chloride antiport and the combined action of a sodium proton exchanger, you basically reabsorb sodium chloride together, and the anion, which could be formic acid and many other things, essentially just recirculates. This is an additional mechanism. And as I mentioned in the previous slide, since you have chloride accumulated, you develop a driving force for chloride, a chemical driving force. So chloride will diffuse through these gap junctions, the tight junctions, sorry, and the lumen will be slightly positive. And the slightly positive lumen will actually drive the absorption of sodium. So eventually sodium and chloride will go through here, these tight junctions, adding to the reabsorbed sodium. So this is a lot of different mechanisms. And the bottom line is that by the end of the proximal tubule, you will have more or less 2/3 of the sodium and chloride reabsorbed, and water will follow. And so the tubular lumen, tubular fluid, stays isosmotic. That was the proximal tubule. Now we come to the loop of Henle, the thin descending limb and a thin ascending limb. Not much active transport happens there. Those parts will be somewhat important for the water balance, but we skip them. They don't do much active transport. The cells are thin, not much mitochondria. So you can forget those for the sodium balance. But now we get to the last segment of the loop of Henle, which is the thick ascending limb. This is a very important segment for many reasons, including sodium reabsorption. And what you have here is the famous sodium chloride potassium cotransporter, which is sensitive to furosemide. Furosemide is the most potent diuretic. The reason it is the most potent diuretic because it inhibits the sodium chloride potassium transporter, and this segment of the nephron reabsorbs 25% of sodium chloride via this single mechanism. And if you block that, you can have a very profound effect on sodium balance. So what you need to know about this transporter is that it has to have all four of these ions for it to catalyze this transport. So it doesn't work if you don't have all four ions, which is 1 sodium, 1 potassium, 2 chloride. If, for example, you run out of potassium, it stops working. And the consequence of that is the extracellular sodium concentration is high, 140 millimolars. And you have something around that in the tubular fluid also, because even you remove a lot of it, water followed. But the potassium concentration is very low. It's only around 4 millimolar. So what happens is this transporter is very efficient. It's just going to clear out sodium. And eventually, at one point, the lumen is devoid of potassium, you run out of potassium, and the whole thing stops. So for it to be able to continue to work, it needs a counter transport, and there's a potassium channel here. Basically, what it does is once you run out of potassium, potassium will leave the cell and replenish the potassium in the lumen. So essentially what happens is potassium is basically recirculated here in order to allow sodium reabsorption. The consequence of that is that this guy, the furosemide sensitive transporter, this is electroneutral. 2 positive, 2 negative charges, no change in charge. Whereas this one, this apical potassium channel, it's an electrogenic transporter, which means once a potassium diffuses through it, there's no countercharge, so it's going to make the lumen positive. And the consequence of the lumen positivity is that this positive, lumen positive transepithelial voltage difference is going to be a driving force for transcellular reabsorption of sodium, potassium, calcium, and magnesium. It's especially important for calcium, and this is one of the reasons why if you take furosemide, it slows down this process, and it doesn't only make you lose sodium, but it also makes you lose calcium because it slows down this mechanism. So this is overall the mechanism. And one more thing to mention, which will be important at the water lecture, is that the water permeability of this segment is very low, which means the tubular fluid will become hyposmotic because you remove salts, but water will not follow. And the mechanism and the consequence of this will come back next week when we discuss water balance. Now, we got to the early distal tubule. This is the simplest one. There's no complicated recirculation. It looks as if an engineer designed it. So it has a sodium chloride cotransporter, which happened to be blocked by thiazide diuretics. It's a different group of diuretics, not as efficient as furosemide because this segment simply reabsorbs less sodium chloride. So thiazide diuretics block this sodium chloride cotransporter and makes you lose sodium and chloride. And this segment is also impermeable to water. Again, the importance of this will be discussed in the water balance lecture. And the final segment with yet another mechanism for sodium reabsorption is the late distal tubule and the collecting duct. What you have in this nephron segment is 2 different kinds of cells, the principal cells and the intercalated cells. And we're only concerned at this point with the principal cells, which are important for sodium reabsorption. What these cells have is this apical sodium channel, which is called ENaC, it stands for epithelial sodium channel. It's a channel, so it's electrogenic. It only transports sodium. And this one is blocked by amylorides. Amyloride is another diuretic, which acts differently from the previous two that we discussed, or three. And what happens here is because the ENaC channel is electrogenic, you have a negative 40 millivolt transepithelial voltage difference because this guy is electrogenic. And what happens is this lumen negative transepithelial potential does two different things. One, it's a good driving force for chloride to be reabsorbed paracellularly. And also, this contributes to the driving force of potassium secretion here, which usually goes hand in hand with sodium. So when sodium is reabsorbed, this cell secretes potassium. And the importance of this will be discussed at the potassium lecture. As I said, everything is connected to sodium in kidney physiology. So the rest of the lectures, we will always come back to the sodium lecture, and you will see the connections between the different topics. So this is the segment where aldosterone, which is the key regulator of sodium balance, acts. And basically what aldosterone does, it simply speeds up this principal cell, reabsorbing more sodium and secreting more potassium. And this is the segment where the water permeability depends on ADH. This one will be discussed at the water balance lecture. And just one or two sentences about the intercalated cells. These intercalated cells are important for acid-base balance. They are in the same nephron segment. And one thing to remember is that this lumen negative transepithelial potential does actually influence the intercalated cells also. So even though the function of aldosterone is to stimulate the principal cell, what happens is when you have too much aldosterone, as a side effect, it will also stimulate the intercalated cells because this will be more negative. It will secrete more protons. So hyperaldosteronism have consequences not only in sodium balance, but also in acid-base balance. And the reason is most likely this lumen negative potential difference. So these were the mechanisms. It's dense. You really just need to memorize these. But it's really important information because everything else builds on it. Plus, these are all targets, or these transporters. Most of them are targets for clinically used compounds which act as diuretics. And do you have a question?
All of these transporters, or several of these transporters, are creating one genetic potentials. Why? How can they be different if one side just flows? All the different ions come together, right? So how can a 50 millimole genetic potential be directed into a 40 millimole genetic?
Yeah, very good question. So what usually the question becomes, what's primary and what's secondary? So here, the primary thing that happens is sodium reabsorption. You have still plenty of sodium left here. So that's the primary thing that happens. And therefore, this potassium, if this potassium channel wasn't here, then this would be even more negative. I think that's kind of the way to put it. Does it make sense? Yes, this decrease. Yes. So basically, this is negative 40 driven by sodium. If this wasn't here, this would be more negative. In other words, this is what you said, that flowing of this potassium constantly decreases this. So this negative 40 is a product of the balance between the sodium channel and the potassium channel. So this is just a summary of all the transporters that we discussed. Again, the proximal tubule reabsorbs 2/3. You have all these different sodium coupled mechanisms. The thick ascending limb has the furosemide sensitive transporter. The early distal tubule, the thiazide sensitive, and the late distal tubule, the collecting duct, has the amiloride sensitive ENaC channel. And so now we get to the regulation of sodium reabsorption. The effectors are most importantly the renin-angiotensin 2 aldosterone system. And you also have the atrial natriuretic peptide and the sympathetic nervous system. And if you remember the previous lecture, these are pretty much the same factors that regulate renal hemodynamics and glomerular filtration rate. So the players will be a repetition, but the effects, we're going to basically extend what we learned at the glomerular filtration rate to somewhat more specific effects to sodium, and we're going to reiterate certain factors that we already discussed in the previous lecture. So the key point of regulating sodium, as I tried to emphasize several times, is to keep the extracellular fluid volume in the normal range. And the reason for that is basically to regulate your blood volume, or in other words, to regulate your effective circulating volume. So sodium balance, really what it does, it regulates the effective circulating volume. That's why it's important to regulate it. What is an effective circulating volume? It's basically a measure of how efficiently the efficiency of tissue perfusion by blood. It's not something that can easily be measured, but it's a volume, and as such, the higher the volume, the higher the pressure is. The sensors that regulate are generally baroreceptors. You have them in the granular cells of the juxtaglomerular apparatus, as you will see in the next couple of slides. This is where renin secretion is regulated. You have them in the low pressure system, in the pulmonary veins and the atria. This is where ANP is regulated, and to some extent ADH, which is primarily a water regulatory hormone, it also contributes to volume regulation, as you will see next week. And you also have the high pressure system, the carotid sinus and the aortic arch, which regulates the sympathetic system. And the homeostatic effectors, short term, your heart and blood vessels. You had this from cardiovascular. If you lay down, if you stand up quickly, your blood pressure drops, but the counter-regulatory mechanisms quickly make your heartbeat faster and your vessels constrict. But the long-term regulator really is the kidney, which will determine on the long run how much volume you have by regulating changes in sodium excretion. So the renin-angiotensin 2 system is the major salt and volume conserving system. If you remember this fact, if you have a question, if you don't remember the details, if you think of the big picture, what is it that will retain sodium? In many cases, you can actually predict what the effect of this system is. So it's good to keep in mind the big picture, that this is the major salt conserving, sodium conserving system. So the first step is that renin, which is a proteolytic enzyme, is produced by the granular cells of the juxtaglomerular apparatus, which is basically specialized smooth muscle cells in the afferent arterioles. And this, the primary factors that stimulate renin release are a fall in the blood pressure. These granular cells are basically baroreceptors. So if your blood pressure here is lower, that's going to turn on renin. That's the primary effect. That's a primary stimulator. And you also have a little bit of a crosstalk from the sympathetic system. So if your sympathetic outflow is low, then your renin is not as high as if your sympathetic outflow would be high. So if your blood pressure drops, the sympathetic will turn on your heart, but it's also going to crosstalk to the renin-angiotensin 2 system. And finally, you also have this sodium and chloride delivery sensing system in the macula densa, the same system that regulates GFR. The same system also sends a signal to stimulate renin if your sodium delivery here is low, to basically increase the sodium reabsorption. So inhibitory factors, you have a feedback regulation, inhibition by angiotensin 2. You have inhibition by the atrial natriuretic peptide. Again, as I said, the renin-angiotensin 2 system is the major salt conserving system. AMP will be the major salt disposing system. So it's basically antagonizing the renin-angiotensin 2 system essentially at every step, including here at the renin secretion. And you also have, if your blood pressure is high or your increased sodium chloride delivery to macula densa, that will also inhibit renin secretion. So now this slide shows quite a bit of antihypertensive pharmacology, as you will see. These are the specific steps of how angiotensin 2 is generated and broken down. And again, this has significant pharmacological relevance. As I mentioned, renin is a proteolytic enzyme which cuts off a 10 amino acid peptide, angiotensin 1, from this larger plasma protein called angiotensinogen. And once the angiotensin 1 is liberated, it's not really yet the active compound. You need a second enzyme, which is called the angiotensin 2 converting enzyme, to cut off an extra amino acid from this angiotensin 1, and it will basically form angiotensin 2. And angiotensin 2 is really the active compound. This is the one that acts on various angiotensin receptors. This is what constricts the efferent arterioles. This is what stimulates aldosterone, et cetera, et cetera. So the angiotensin 2 converting enzyme is a pharmacological target. You have inhibitors. They're called angiotensin converting enzyme, or ACE inhibitors. The prototype is captopril. Many drugs that end with pril, they are usually ACE inhibitors. And so the idea is simple. If you block this enzyme, you generate less angiotensin 2, and therefore your blood pressure will decrease because you don't have angiotensin 2, which both stimulates sodium reabsorption. It's also a vasoconstrictor. The interesting part is that the ACE enzyme doesn't only convert angiotensin 1 to angiotensin 2, but it also breaks down bradykinin, which is a peptide that has several different effects. It generates, it plays a role in pain, but it also plays a role in cough, and it also happens to be a vasodilator. And quite interestingly, the angiotensin 2 converting enzyme, for whatever reason, is mainly in your lungs. And what happens, it breaks down bradykinin there. And so if you block it, you don't break it down, so you have more bradykinin, you have more vasodilation, which is good. You're taking the ACE inhibitors to start with because you're hypertensive. So if you have more vasodilation, that's good. What is not that good is that it also generates cough, and because bradykinin also something that induces both pain and cough. But the major side effect of these drugs, in some people at least, is coughing. And it's logical because these ACE inhibitors block the breakdown of bradykinin. If you have more bradykinin, cough more. And that's the major reason why later specific antagonists of the AT1 angiotensin 2 receptor were also developed. The prototype was losartan. And if somebody takes ACE inhibitors and has this side effect of cough, they can be switched to angiotensin receptor antagonists. And finally, even more recently, inhibitors of renin have also been developed. So these are also an option to block this pathway. This is the pharmacology of the renin-angiotensin 2 system. The next slide, this is a slide that I only introduced last year. this just basically repeats the previous slide. What it also has is the ACE2 enzyme, which converts angiotensin 1 into this other form of angiotensin and converts angiotensin 2 to this angiotensin 1 to 7. And the reason why ACE2 is important these days, because it happens to be the receptor for the SARS-CoV-2 virus. And this is not inhibited by ACE inhibitors. Initially, people were enthusiastic. Maybe we should just take ACE inhibitors to treat COVID. I don't think it works. And also these products of the ACE2 enzyme don't act on actual angiotensin 1 receptors. They act on something else called the MAS receptors. I don't think that physiology is very well understood. I just inserted this slide for the sole reason that the COVID virus binds to pulmonary cells through this, this relative of the ACE enzyme. Now, if we go back to the effects of angiotensin 2, we discussed in the previous lecture that it's a vasoconstrictor. It mainly constricts the efferent arterioles. It mainly increases the resistance of the efferent arterioles, and it will help sodium and fluid reabsorption by hemodynamic effects. And the way it works is because of the efferent vasoconstriction, the renal plasma flow will go down, but the GFR will go slightly up, or it doesn't change. And the end result will be that the filtration fraction, the fraction of GFR divided by RPF, will increase. And if your filtration fraction is higher, then it will increase the protein concentration in the peritubular capillaries, and that increased oncotic pressure in the peritubular capillaries will be a driving force for moving the reabsorbed fluids back to the general circulation. This is just a cartoon basically showing the idea that after you reabsorb fluid into the interstitial compartment, you still need to move that into the peritubular capillaries. And if this fluid movement is not very efficient, some of the fluid can actually leak back to the tubular fluid. If this is efficient, then you clear out this fluid and make the reabsorption more efficient. So angiotensin 2 has this hemodynamic effect to help sodium and volume reabsorption. An additional factor in this hemodynamic effects is that, of course, if you constrict either one of these arterioles, the hydrostatic pressure will also drop in the peritubular capillaries, and that also adds to the Starling forces, moving fluid back to the general circulation. So these are hemodynamic effects that we discussed already in the previous lecture. And now there's specific effects for sodium. So angiotensin 2 has some tubular effects in the proximal tubule and the thick ganglion, stimulating the sodium proton exchanger. You already discussed that it's a general vasoconstrictor. One of the reasons why you have hypertension, if your renal arteries are stenotic or constricted, is that you generate angiotensin 2, and it will constrict your vasculature. Angiotensin 2, interestingly, if it's stimulated, also makes you thirsty. And I made a big case on lecture 1 that the sodium content is the most important regulator of volume, but of course volume is given by water. So one thing that if you have more sodium, you attract water from the intercellular fluid, but eventually if your sodium content is up, you're not only going to be hypervolemic because suck out water from your cells, you're also going to be hypervolemic because you're going to drink. You're going to drink because of angiotensin 2. You're also going to drink, as you will see next week, that if you're hyperosmotic, that's itself a stimulation for drinking. So that's this is an additional mechanism how the sodium content will increase eventually your volume, not so much your sodium concentration. And finally, probably the most important effect of angiotensin 2 is to stimulate aldosterone production. So aldosterone is a steroid hormone. You also learn about this in the endocrine lectures. It's produced in the outer layer of the adrenal gland, the zona glomerulosa cells. It is regulated by angiotensin II. It's the final step of the renin-angiotensin II aldosterone system. And it's also regulated by plasma potassium concentrations. As you will see on the potassium lecture, and as already sort of mentioned it, aldosterone also regulates potassium. And potassium stimulation of the aldosterone secretion is part of this potassium regulatory feedback loop. And the effects of aldosterone is basically to speed up the principal cell by, it's a steroid hormone, so it mainly acts transcriptionally. It will increase sodium, potassium ATPase and ENaC generation, and the end result will be sodium is reabsorbed more, potassium is secreted more. And as I mentioned earlier, because of the intercalated cells in the same nephron segment, it will also stimulate the secretion of protons. And now from the renin-angiotensin II system, we can move to the sympathetic nervous system. So norepinephrine, the sympathetic neurotransmitter, constricts both the afferent and the efferent arterioles, and it will eventually decrease the renal plasma flow, and it's going to reduce the sodium excretion through hemodynamic effects by decreasing the hydrostatic pressure in the peritubular capillaries and reducing sodium delivery. As I mentioned earlier, it also stimulates renin release, which is basically a crosstalk between the two systems. And finally, it also has direct tubular effects by stimulating sodium reabsorption in proximal tubule cells, stimulating the sodium proton exchanger and the sodium potassium ATPase. And finally, natriuretic peptides. The atrial natriuretic peptide is the major sodium disposing system. It's in the atrial myocytes. When the atrial pressure increases, it signals volume overload. So it basically increases sodium excretion, partly through hemodynamic effects, as we discussed before. And it also inhibits renin release. It inhibits aldosterone secretion directly. It also has direct tubular effects, inhibiting sodium transport in the inner medullary collecting duct. It's a vasodilator. Just as I said with the renin-angiotensin II system, if you remember the big picture that AMP is a sodium disposing system, you can make a good educated guess of what it does to each and every step, because each and every effect serves the final goal of disposing sodium. Interestingly, AMP signals by cyclic GMP, which is not very common. The only other thing I know of that uses it is nitric oxide, which is endothel-derived vasodilator. And we're almost done. This is the summary of diuretics, as we discussed. Carbonic anhydrase enzyme inhibitors like acetylzolamides reduce bicarb reabsorption together with sodium in the proximal tubule mainly. Loop diuretics inhibit the sodium potassium 2 chloride transport in the thick ascending limb. Thiazides, the sodium chloride co-transporter in the distal tubule, and ENaC inhibitors like amiloride block the ENaC channel in the late distal tubule and in the cortical collecting duct. What I did not mention is you can also use antagonists of aldosterone. One of them is spironolactone, which have very similar effects to amiloride, because instead of blocking directly the channel, they block the effect of aldosterone, and the effects of amiloride and spironolactone are quite similar for this reason. And this concludes this lecture. If you have any questions, you can stay. I can put on my mask. And if not, you can email me. And otherwise, see you next week. Actually, see you on Friday on Zoom.